Let's talk about core concepts. When we think of humans, or more broadly animals, we tend to think of them as the whole organism. However, we are in fact a collection of highly specific organ systems. These organ systems are able to carry out specific tasks due to the function of each of the individual organs that contribute to the organ system, such as the breakdown of food into molecules small enough to be absorbed into the blood via the digestive system. Each organ within the organ system has a specific role which can only be performed with the right tissues. The primary role of the small intestine is to absorb the molecules that the digestive process in the stomach have liberated from food. Tissues are adapted to their specific tasks through the specific nature of its collection of different cells, such as the many convolutions of the intestinal epithelium, which provide a large surface area for absorption of nutrients. Different cells have unique functions as a result of specific organelles, which allow them to perform their roles, such as selective absorption of nutrients, which occur through the intestinal epithelial cells. Selective absorption is determined by the exact molecular makeup of the lipid bilayer. The combination of atoms in the phospholipid molecule create kinks and bends within the bilayer, which alter the fluidity of the bilayer and therefore the ability of molecules to pass through. Different atoms are able to bind together in different ways, forming variable numbers of chemical bonds, which alter the way in which the final molecular structure appears. The cell is the smallest unit of life. While they look quite inert when examined through a microscope, they are in fact a bustling metropolis with a border and many individual units which have specific tasks and functions. Common units within the cell, known as organelles, are the nucleus, which holds all the data that encodes life, the mitochondria, which are the power stations, the ribosomes for making proteins, the endoplasmic reticulum, both rough and smooth, for modifying proteins, the Golgi apparatus or body for packaging proteins, the centrioles from which the cell scaffolding originates, and the plasma membrane, which holds everything important in and everything else out. Put each of these organelles into your Moodle glossary and find out some more details about what they do. While cells all have the same basic structure, they do not all look the same. Specific cells have shapes that are designed to fit the task. Sperm cells have a long flagellum to allow them to swim. Red blood cells are designed to have a high surface area to allow maximal oxygen uptake in the lungs. Neurons have long extensions so they can communicate with other cells in far-lying locations. The ovum is the largest cell in the body, as it essentially holds the ingredients of life except for half the DNA which comes from sperm. And muscle cells have adapted long fibrous structures that allow them to be effective pulleys. As with the whole organism, cells also require nutrients and other important information molecules to pass in, and waste products and other secretions to pass out of the cell. There are four main mechanisms of transport across the cell membrane. Passive diffusion allows a very small number of molecules to pass directly into the cell by moving from areas of high concentration to areas of lower concentration, slipping between the phospholipid molecules. But as the bilayer is essentially made up of fat or lipids, and the majority of compounds moving around are either water-soluble or too big to squeeze through, this is a limited means of entry, hence why the lipid bilayer is referred to as selectively permeable. Another means by which the bilayer is selectively permeable is through protein channels. These proteins are embedded in the bilayer spanning both phospholipid layers. Small water-soluble molecules are able to pass into the cell via these channels, which are often gated to only open in response to a specific signal. Again, molecules travel down a concentration gradient from high to low concentration in an effort to equalize the concentrations on both sides of the bilayer. When molecules have to move against the concentration gradient, then energy is required in the form of ATP. These specific molecules bind to a channel protein, and the binding of ATP and breaking of a phosphate bond causes the protein to change shape, allowing the molecule to be released into the region of higher concentration. Really, really big things which cannot pass through a channel protein can be brought into the cell by a process known as endocytosis. One special version of this is phagocytosis, where a bacteria or virus or disease cell is taken into a cell for destruction. In the process of phagocytosis, the lipid bilayer forms a pocket around the cell or bacteria it wants to engulf. The bilayer then pinches off to form a vesicle inside the cell. Lysosomes can then merge with this vesicle and release digestive juices to destroy the contents. Releasing compounds like hormones from the cell can also be achieved through vesicles, with the hormone-containing vesicle moving out to the lipid bilayer from the Golgi apparatus, fusing with the bilayer and allowing its contents to be released into the interstitial space. This is a process known as exocytosis. There are four main tissue groups which combine to create the organs of the body: muscle tissue, nervous tissue, connective tissue, and epithelial tissue. Many different cell types come together to make these tissues, which have specific functions. Add these tissues to your Moodle glossary and write definitions which include the main function of the tissue. For example, muscle tissue's primary function is to cause movement. Can you find some specific examples of each type of tissue? There are 11 major organ systems. In your fact-gathering mission, you'll be investigating what each of these systems do and which organs belong to each group. In no particular order, we have the nervous system, the respiratory system, the cardiovascular system, the blood and lymphatic system, the muscular system, the skeletal system, the endocrine system, the renal system, the digestive system, the skin or integumentary system, the male reproductive system, and the female reproductive system. In all the systems, multiple organs come together to perform the primary system's role, and all four tissue types are required to create functional organs. Which brings us to the final key physiological concept for this unit: homeostasis. It's all about balance. As animals, we are very sensitive to change. Just as 18 degrees is starting to feel cold, and 23 degrees is getting a bit too hot, for those of us who grew up in the North Island at least, our cells, tissues and organs have a very narrow range of conditions within which they work optimally. Outside of this range, things start to go wrong and illness or injury can result. As we live in a dynamic environment where things are changing constantly, being unable to adapt to changes as they occur would not result in very long species survival. Animals have in fact done quite well, and it's because of the concept of homeostasis. Homeostasis is primarily controlled by feedback loops, both negative and positive feedback loops. And once you get your head around these, then you will have a good idea of how most systems adapt to change. A typical negative feedback loop, there are 5 separate events. 1. Something changes. 2. A sensor senses a change. 3. A message is sent to the central processing unit. 4. The central processing unit sends a message to the effector to make changes so that 5. Conditions are returned to within the optimal level. An example of such a negative feedback loop is the insulin response which occurs when you eat something rich in carbohydrates. Following ingestion of a carbohydrate-rich meal, your digestive system breaks the carbohydrates down into simple sugars, primarily glucose. Glucose is absorbed through the intestinal walls into the blood. An increase in blood glucose is sensed by receptors on beta cells in the pancreas, which then send a message to release insulin into the blood. Insulin stimulates fat, muscle, and liver cells to take glucose out of the blood and store it as lipid or glycogen for later when blood glucose levels are low. Thus, by sensing chemical messages and sending other chemical messages, blood glucose homeostasis is restored. There are many cycles like this which occur in the human body, and one of your tasks will be to investigate these. Our final bit of information we need to dig into is terminology. More terminology, you might ask? Yes. Unfortunately, anatomy is all about the terminology, and to be able to be a medical interpreter, you need to understand these. When talking about the human body, we always refer to it with respect to the anatomical position. There are some key features of the anatomical position to note. See if you can figure them out. Once we are in the anatomical position, then we can start to refer to different parts and planes of the body. There are 3 planes: sagittal, frontal, and transverse, in which we can view and dissect the body. Anterior and posterior, medial and lateral, dorsal and ventral, and proximal and distal allow us to locate areas of the body with respect to others. While the terms flexion and extension, abduction and adduction, internal rotation and external rotation refer to movement of segments via joints around the 3 anatomical planes. You will note that I've presented these terms as pairs because they are opposite. For example, extension is the opposite of flexion. While this overview of topic 1 might seem terrifying, it is designed not to bombard you with facts, but to provide you with terminology to aid you in searching for information, and to give you a point from which to start to investigate the fascinating world that is your body.